Hello and welcome to Invertebrate Paleontology and Paleobotany at Utah State University. This is lecture 14 where we'll discuss the question, what evidence do we have for the earliest single-celled life in the fossil record? Geologists split the history of Earth into two major periods of time. The Phanerozoic, the time of life, where fossils are abundant and well represented in the rocks, and the Precambrian, a vast length of time occupied by single-celled organisms and rare tiny fossils and stromatolites. As a young geologist, I often viewed the Precambrian as a no-man's land of fossils, a vast series of hard rocks with no discernible evidence of life. However, since then, numerous discoveries are pointing to a vast period of Earth's history in which life at the single-cell stage was playing a major role in the transformation of the planet, and evidence of these tiny organisms are being rapidly discovered in the Precambrian rocks. The Precambrian today is divided into the Hadean, the Archean, and the Proterozoic. Each period of time demarcates a new transition on the planet. The Hadean was a period of 600 million years, about equal to the length of time of the Phanerozoic, from about 4.6 to 4.0 billion years ago, when the Earth was cooling down. As such, Earth was a bit too hot for life, as much of the surface was covered in molten lava and the consistent bombardment of meteorites. It was during this time that the Moon formed. No rocks have been found from this period, and geologists must study moon rocks, or meteorites, to peer into insights of this time period. We can assume that life was not yet around, but this is from lack of evidence. The Archean is a time period from 4.0 to 2.5 billion years ago, or about 1.5 billion years, in which life was getting started. It's the age of bacteria, of prokaryotic life, single-celled tiny organisms that leave behind a fossil record of bacterial mats called stromatolites. Now, stromatolites are these large accretionary structures thinly layered by the acquisition of detritus on a sticky surface of cyanobacteria filaments, growing in shallow marine waters and photosynthesizing. The oldest stromatolite dates from 3,524 million years ago in the early Archean. The oldest stromatolite comes from the Dresser Formation of Australia. These stromatolites differ from younger stromatolites in the way that they were preserved. Geochemically, these stromatolites are composed of pyrite, which is thought to replace the original matrix of carbonate. Pyrite is iron and sulfur, and some suggested that these early bacteria utilized sulfur like some deep-sea vent bacteria do for chemosynthesis. The regional geology suggests that these stromatolites lived in a collapsed caldera, suggesting that these bacterial mats lived in a volcanically heated, very warm hot waters, much or similar to the Yellowstone caldera today, but one which had been flooded by ocean waters. So imagine a shallow, hot marine setting. The presence of pyrite and hematite suggests that the atmosphere lacked oxygen or was severely depleted of oxygen. Instead, it was likely composed of nitrogen, carbon dioxide, methane, and ammonia. Despite a more dim early sun providing sunlight, the Earth was actually much warmer due to the thick atmosphere and internal heat from the relatively recent formation of the Earth. In the nearby Panorama Formation, dated slightly younger at 3,460 million years ago, are unusual, strange, conical stromatolites. One of the challenges of these early stromatolitic fossils is whether they really are fossilized evidence of life, or were somehow produced by inorganic geological processes such as deformation of bedding or faulting. One technique is to look at the carbon isotopes found in stromatolites. Carbon tends to bond to either oxygen or hydrogen. The most common carbon-oxygen molecules are carbonate minerals such as calcite, while the most common single carbon-hydrogen molecule is methane. Methane, or natural gas, unlike carbonate minerals, is volatile and easily combusts in the presence of oxygen. Hence, because of this exothermic reaction, energy is often required for the formation of these bonds, and most carbon-hydrogen molecules are formed by organisms or life forms. Carbon-hydrogen bonds favor lighter carbon isotopes, or C12, while carbon-oxygen bonds favor higher carbon isotopes, C13. Measuring the ratio of lighter carbon isotopes, or C12, to heavier carbon isotopes, C13, gives you a relative way to measure how organic a fossil might be. If carbon formed from the polymers of organic matter, such as DNA, RNA, as well as proteins, it will have a more negative ratio of carbon-13, while carbon formed from bonds with oxygen to form carbonate minerals will have a more positive ratio of C13. Using these isotopes, these early fossils show negative ratios of carbon-13, indicating that they likely came from organic molecules rather than inorganic carbonate. This technique has been used to extend the record of life back another 300 million years, but the technique is not without problems. An early atmosphere that lacked oxygen would mean that inorganic methane would have a much longer residence time in the atmosphere, and hence you need to compare the ratio of the fossils to the background matrix of the rocks. It's hard to envision a mechanism that would trap inorganic methane or other carbon-hydrogen molecules in a rock. This isotopic evidence suggests that these early Archean fossils are indeed evidence of Earth's earliest life forms. Stromatolites occur throughout the Archean, but paleontologists have also been looking for other evidence of early microscopic life. Microfossils have also been found in the same units as stromatolites. The most famous of these are the fossils from the Gunflint Chert, dated at 1.88 billion years ago, from a series of banded iron formations in Minnesota and Ontario. Discovered in the 1950s, the unique formation preserves a high diversity of microscopic single-celled organisms within a Chert matrix. Two types of forms exist: spherical forms as well as filament forms, long strands that closely resemble cyanobacteria. These fossils are from the Paleoproterozoic, the first 900 million years of the Proterozoic. It was likely that during this time the first eukaryotes evolved. As we head further into the Proterozoic, in the Mesoproterozoic, we find more evidence of forms of microscopic fossils, such as these from the Bitter Creek Formation of Australia that date to around 850 million years ago. These include colonial Cocoolithian forms where the cells group into small balls of cells. There's also strands of cells similar to the ubiquitous cyanobacterial forms. During the Neoproterozoic, around 1,000 million years ago, or a billion years ago, we get the first archeans. Archeans are the strongest proof of the origin of eukaryotic life, as they resemble large carbonate-composed cells found in siltstones and shales. Archeans are thought to be algal cysts, which represent a two-stage life cycle in these early eukaryotic organisms, similar to red algae today. Cysts would be coated with a protective skeleton of carbonate, or maybe possibly silica, which allows them to survive during these adverse environments, such as desiccation from dropping sea levels. The organism would lay dormant on the floor of the ocean or sediment until the environment would improve and then sprout out various cells that would float in the water photosynthesizing, then mate and settle out of the water as cysts. The planktonic cells would be gametes, while the cyst would be the zygote part of the life cycle. The cysts preserve, but the planktonic forms do not, since they lack hard protective shells. Archeans become common in the Neoproterozoic from about 800 million years ago into the Phanerozoic, and can be used for biostratigraphy in the Neoproterozoic. At 630 million years ago, we have the last period of the whole of the Precambrian, the end of the Neoproterozoic, the Ediacaran, following the Great Snowball Earth period of the Cryogean, in which we start to get the first multicellular life. The first multicellular life appears in the Ediacaran of Australia, but the recent discovery Ediacaran multicellular plant life in China also points to the collective emergence of both multicellular plants and animals during this period of time. The first period of the Phanerozoic, the Cambrian, sees a major increase in both multicellular plants and animals. In the next series of lectures, we'll examine the diversity of multicellular animals, starting with one of the most primitive groups, the sponges. Thank you for watching this lecture. If you're interested in taking a class at Utah State University in the Geology Department, check out the geology website at geology.usu.edu. And if you're interested in my own research and who I am, check out my website at benjamin-berger.org. Thanks for watching.